Hello friends, I am Archana Hegde and today's topic is staining. Staining is a very beautiful technique or a method of microbiology where the microorganisms are stained and they look very beautiful under microscope, and microorganisms so tiny creatures which cannot be seen under naked eye, so it is seen under microscope in different resolutions 10x, 40x, 100x or oil emulsion field. So coming to staining is a technique used in microscopy to enhance contrast in the microscopic image. This means to enhance the color of which cannot be visible if it is not stained, or it may be transparent. So to enhance its visibility we color it or we stain it to see under microscope, which is a microscopic image what we get under microscope. Stains used to highlight microorganisms or biological tissues for viewing with the help of microscope. Aims of staining: to increase visibility and contrast. This means to see the visibility or to view the microorganism under microscope. This is also used to study the morphology. Morphology means cell structure, cell membrane, nucleus, etc., which is inside or outside or surrounding the microorganism under microscope. Third thing, to detect extracellular and intracellular component of microscope. It is similar to cell wall, cytoplasm, cytoplast, etc. To help in the primary level of diagnosis, in hospital level or in the diagnosis field, this staining technique is very important, and this is the preliminary or first step used in the diagnosis laboratories. Coming to the different type of staining in bacteria. In bacteria, there are three main types of staining, namely simple staining, differential staining and special staining. In simple staining, it is divided as positive and negative staining, where the dyes are positively charged are called positive staining, whereas the negatively dyes are called negative staining. Positive staining contains of methylene blue stain, dilute carbylfuscin. Negative stainings are Indian ink method or Nigrosin method. Coming to the differential staining is a method where one or more microorganisms can be stained together to view in a same microscopic field, and also different stains can be used, like primary stain or secondary stain. This contains Gram stain and Zn staining, which is also called Zil-Nelson staining. Coming to the special staining, there are many special stains which are used for only for particular purpose, like to stain spores, capsules, bacterial cell wall, bacterial flagella, and also for different microorganisms. Different stains are used for spirochetes, Mycoplasma, Rickettsia and Chlamydia. Different stains are used, and also there is one more stain called Albert stain. Coming to the simple staining is a technique used in bacterial smears with single reagent, nothing but single stain or single color. It may be a positive stain or it may be a negative stain, but only one stain is used. Commonly used simple stains are basic stains, methylene blue, crystal violet and carbylfuscin. In this also, stains are divided into basic and acidic, which are basic and some are acidic in nature. Coming to the principle, basic stain with chromophores are used, like chromophores which are having the content to attract bacteria. If they are positively charged, they attract negatively charged bacteria. If they have a negative chromosome in them, they attract the positive charged bacteria. Basic stain with chromophore are used. Bacterial nucleic acid and certain cell wall components carry a negative charge that are strongly attracts and bind to the cationic positive charge chromosome, imparts same color to the bacteria, to all the bacteria where it is attracted. Procedure: take a slide and take a loop full of culture and smear it over that and stain it for 3 minutes and wash with water and dry clean. For tissues, it takes 5 minutes. Your observations are that blue color is spherical cells. Blue colored colony which are elongated, rod shaped are bacilli which can be seen. Blue colored cells per cell. Why this blue color here? Because maybe you are using a simple strain called methylene blue. So it tends to give a blue color. So coming to this image here, the left side image is shown to be rod shape, which is the second one, elongated rod shaped cells which are bacilli, which are in pairs, chains or in singles. Coming to the right side image, these are the round shaped or spherical shaped cells which are also called as cocci. Cocci will be in pairs or in single or also in chains. Coming to the uses, here in this image we can see both stains are used, like simple stain scenes for rod shaped bacilli which are in chains, and these are the cocci. So here for the diagnosis purpose it is used. Lofer methylene blue demonstrates morphology of polymorphs, lymphocytes and other cells more clearly. Polymorph methylene blue demonstrates presence of bacteria in smear, morphology of polymorphs or lymphocytes. To demonstrate acidic capsular material of the anthrax bacilli. Coming to the negative staining. Negative staining is very simple and very nicely imaged microscopic staining technique, where in this the background gets stained, organism remains colorless. That means whatever stain we are adding, the microorganisms are negatively charged. So when the stain is also negatively charged, negative repel to each other. So instead of coloring the microorganism, the circumference or the background of the microorganism will be colored, so that the organism cease to be very it is like colorless and it seems like a night sky having stars in background black sky. We can see a microorganism as stars and background will be totally black or brown in color. These stains are acidic stains, for example Indian ink and nigrosin. Coming to the principle, these are acid dyes. Negatively charged chromosomes will not readily combine with negatively charged bacterial cytoplasm. Instead forms a deposit around the organism, leaving organism itself colorless. In this, two types are there, namely Indian ink and nigrosin. Indian ink is a method. Mix small quantity of Indian ink on a slide with the culture or material containing bacteria. Make thin film with a loop and allow it to dry. Do not heat fix in a negative staining. Here bacteria are as clear transparent objects on a dark brown background. Coming to the nigrosin method, place a drop of dye on a glass slide, mix the culture or material containing bacteria with the dye. Make a thin film with help of loop, allow it to dry. Bacteria as transparent objects against absolute homogeneous black background. This method is also used for seeing capsulated bacteria or non-capsulated bacteria. The capsules is a very one type of mucus layer which surrounds the bacteria which are capsulated. So the capsules can be seen around the circumference of the bacteria. Coming to the differential staining. Differential staining is a technique helps to differentiate type of bacteria. Here more than one dye are used and bacteria of different types identified simultaneously depending on difference in their staining characters. Like some take basic stain, some take acidic stain, which is also called as primary stain and secondary stain. Secondary stain is also called as counter stain. This is in this two types are there, gram stain and Zaden stain. Gram stain is invented by Hans Christian Gram in 1884. The name grams was given in honor to the scientist who invented this stain, so hence the name gram stain. Very useful stain in microbiology, helps to differentiate or classify bacteria into gram positive and gram negative groups, where in gram positive methylene blue stain will be used, so the gram-positive bacteria takes blue color, whereas the secondary stain are called a safranin stain, which the organism which takes safranin or acetic stain gives a pink color. Coming to the acid-fast stain invented by Paul Ehrlich helps to differentiate acid-fast from non-acid-fast organism. Acid-fast are the bacilli, for example TB, tuberculosis, which is stained in red color, whereas the non-acid-fast are stained as blue color because of the carbon-14 stain used in that. Theories of gram staining. There are many theories, namely cell wall theory, magnesium ribonucleotide theory, cytoplasmic pH theory, etc. One by one we will discuss cell wall theory. Cell wall is the outer membrane of a bacteria. Cell wall of gram-positive bacteria are 40% 40 times thicker than those of gram-negative cells. Hence they are thought to help retain diiodine complex very much. Here gram-positive bacteria and gram-negative bacteria, two types of bacteria are taken, where if in gram-negative bacteria they have a single cell wall, one layered cell wall, but in gram-positive bacteria they may be more than 3, 4, 5, 6 or many more cell wall layers. So it is multi-layered, whereas gram-negative are single layered. So when diiodine complex is used in the gram staining technique, diiodine complex is lost by the alcoholic action or decolorization in gram-negative bacteria, whereas in gram-positive bacteria many layers will be there, the iodine complex will be retained there. So this iodine complex may be retained there, so it may take the gram-negative stain, whereas in gram-negative bacteria it may take gram-negative stain. Magnesium ribonucleotide theory. Compound of magnesium ribonucleotide and basic protein concentrates at cell membrane helps gram-positive bacteria retain the primary dye. Gram-negative bacteria do not possess this substance. The same theory is used here. Cytoplasmic pH theory. The cytoplasm of gram-positive bacteria are said to be more acidic than those of gram-negative ones. Hence dye is said to bind with more affinity to gram-positive cells. Lipid content theory. Cell envelope of gram-negative bacteria contains an additional membrane. Outer membrane contain more lipids than gram-positive bacteria. Coming to the next segment, alcohol or acetone dissolves lipid, thus forming large pores in gram-negative bacteria through which the diiodine complex leaks out. Here the explanation is, after the iodine step the alcohol will be used for decolorizing. So when the decolorizing is done using alcohol, lipid layer in the gram-negative bacteria forming large spores, which forms a large spore, hence the diiodine complex leaks out. So there will be no diiodine complex because of the decolorization. Alcohol or acetone dehydrates the gram-positive bacteria, shrinking the cell wall and closing the pores. Coming to the gram staining principle. Gram reaction depends on permeability of the bacterial cell wall and cytoplasmic membrane to diiodine complex. The permeability is that moving of the object inside a cell wall, or it may be fixed there or it may leak out. Which is leaked out in gram-negative, it may retain in gram-positive. So this importance should be known. In gram-positive bacteria, crystal violet dye iodine complex combines to form larger molecule which precipitate within the cell. But this doesn't happen in gram-negative. Alcohol or acetone mixture acts as decolorizing agent, cause dehydration of multi-layered peptidoglycan of the cell wall, causes decrease of the space between the molecule, causing cell wall to trap crystal violet iodine complex within the cell. Gram-positive bacteria do not get decolorized and retain primary dye appearing violet. Gram-positive bacteria are more acidic, have more acidic protoplasm and hence bind to basic dye more firmly. In gram-negative bacteria, alcohol, being lipid solvent, dissolves outer lipopolysaccharide membrane of the cell wall, which damages cytoplasmic membrane to which peptidoglycan is attached. Diiodine complex not retained within cell and permeates out of it during process of decolorization, takes up color of counterstain and appear pink. Coming to the procedure. Gram staining procedure. First, the methyl smear is done over the glass slide using a loop with 24 or 48 hours culture, and methylene blue stain is added over the slide as you can see in this picture. First, crystal violet or methylene blue will be added. Then wash with the water for after 1 minute. Then add grams iodine for 1 minute. Hold the slide the same for 1 minute and wash with the water. Then immediately decolorize with alcohol like drop by drop, and then again wash with water. Again add a counter stain called safranin, which is pink in color, for 30 seconds to 1 minute. Applications of bacteria. Differentiation of bacteria into gram positive and gram negative is step towards clarification or identification of bacteria. Also, first step towards identification of bacteria in cultures. Observation of bacteria in clinical specimens provide a vital clue in diagnosis of infectious disease. Whether it is a gram positive bacteria or gram negative bacteria can be identified. Useful in estimation of total count of bacteria. How many bacteria are present in this particular slide or in this particular field can be calculated very easily. Empirical choice of antibiotics can be made on the basis of gram stain report. Choice of culture media for inoculation can be made empirically based on gram stain report. Uses. Used in pus specimen. Sputum specimen useful to detect Streptococcus pneumoniae. Stool specimen useful to detect Candida, Clostridium difficile. Urine sample. Presence of at least one organism per oil immersion field correlates with significant bacteriuria, which is less than 10 power 5 CFU per ml. Here CFU is colony forming unit. Coming to the image here, you can see both gram positive and gram negative. Where gram positive takes blue color, here cocci are seen. These two are in chain, this is in paired, and this is very single colony. Whereas gram negative are both cocci can be seen or bacilli can be seen, which are rod shaped, and cocci are round shape. Here these are bacilli. Coming to the acid fast staining. Acid fast staining is a process mainly used for detection of TB. Principle. Acid fastness attributes to the presence of large quantity of unsaponificated wax called mycobic acid in cell wall and intacs of cell wall. Application of heat helps dye to penetrate tubercle bacilli, resist decolorization and confers acid fastness. Here tuberculosis bacilli resist decolorization. So when you pour the alcohol or a decolorizing agent, the decolorization doesn't happen. So hence the acid fastness is seen. Whereas non-acid fast bacilli will not resist this decolorization, hence the blue color is seen. Coming to the ZN procedure in acid fast staining. Make a smear, air dry, heat fix. Flood smear of carbon-fuscin stain. Carbon-fuscin is lipid-soluble phenolic compound which is able to penetrate the cell wall of tubercular bacilli. Steam for 10 minutes. Add more carbon-fuscin stain as needed. Rinse with distilled water. Flood slide with acid alcohol. The acid alcohol contains 3% HCl and 95% ethanol, or decolorize with 20% H2SO4. The waxy cell wall then prevents the stain from being removed by the acid alcohol decolorizer once it has penetrated the cell wall. The acid alcohol decolorizer will remove the stain from all other cells. Tilt slide to 45-degree angle over sink and add alcohol drop by drop until the red color stops remaining from the smear. Rinse with water. And loafer methylene blue stain, which is a counter stain here. This stain adds blue color to non-acid fast cells. Leave loafer stain for 1 minute. Rinse the slide, blow dry, and observe under microscope. This is the microscopic observation where the acid fast bacilli are seen in red color, which are rod shape over this image here. These are the acid fast bacilli, whereas the non-acid fast are taken the blue color, as you can see here. Coming to the special stain, like in special stain, this is the main important staining, Albert staining. Corynebacterium diphtheriae granules made up of polymetaphosphate, which with basic dye granules tend to stain more strongly than rest of the bacteria. Principle: granules present in diphtheria bacilli exhibit metachromasis property, where granules appear in a different color from that of the dye used. Granules appear bluish black color when stained with toluidine blue present in Albert 1 stain. Diphtheria bacilli appear green due to malachite green present in Albert 1 stain. Here, malachite green is also one of the stain. Procedure: cover with Albert 1 stain for 3 to 5 minutes over the culture on the slide. Wash in water and blow dry. Cover slide with Albert iodine and allow for 1 minute. Wash and blow dry. Observation: green colored bacilli show Chinese letter arrangement at angles to each other containing bluish black metachromatic granules. Uses: to stain Corynebacterium diphtheriae. This is one of the technique used in hospital laboratories. As it is seen, the green color bacilli showing Chinese letter here, Chinese letter arrangement at an angle to each other containing bluish black metachromosis. Thank you everyone. This is the staining video, and if you like my video, please subscribe and press the bell button which is beside that. For any comments or any clarification, please comment in the comment box. Thank you.